Transcription. When a eukaryotic cell has a gene turned into a protein, the DNA first unwinds itself and replicates itself. The DNA is joined by an mRNA polymerase, which separates the two double helix DNA into two. One of the strands becomes a transcription unit, and the other strand becomes a template strand of DNA. The promoter is used as a location for the RNA polymerase to go to the DNA, which lets the RNA polymerase start transcription. The terminator is used as a sequence that signals an end of transcription. The initiation of transcription is actually the activation when RNA polymerase attached to the promoter's DNA strand unwinds, and the polymerase starts DNA synthesis at the starting point of the template chain. The elongation that occurs during transcription is when the polymerase moves downstream, elongating the RNA transcript 5 inches to 3 inches. In the wake of transcription, DNA strands form a double helix. The next process is termination. During this process, RNA polymerase II transcribes a sequence, then which codes for polyadenylation signal sequence, which codes for a polyadenylation signal, AAUAAA, in the pre-mRNA. Moreover, at the point about 10 to 35 nucleotides downstream from AAUAA signal, proteins associated with the growing RNA transcript cut it free from the polymerase, releasing the pre-mRNA.
After the end of transcription, changes occur on the RNA. There will be some introns, and there will also be some exons between the introns. However, this experiment is not to see the introns, so we won't talk about the information. Both ends of the mRNA molecule will be altered, but the two ends will be affected with two different things. A 5' tag will be added, and it modifies the pre-mRNA molecule. The poly A tail, a sequence of around 50 to 250 adenine nucleotides, will be added to the pre-prime end of the mRNA molecule. If we did include the introns during this experiment, the introns would be between the following regions. SNRNP will still be used in transcription. It is located in the cell's nucleus and is made up of RNA and protein molecules. A group of tRNA run together along the transcription, and it would combine with each other to form a lysozyme. This lysozyme will interact with peptides along the introns if they exist. Lastly, the reason we would have to use this kind of protein synthesis is so that we can have the mRNA only contain the exons, which is very important.
In between mRNA processing and translation, the amino acid tRNA synthetase joins the amino acid and the tRNA together so that they form a covalent bond attachment. Like transcription, translation is also split into 3 steps: initiation, elongation, and termination. Energy for translation is provided by several GTP molecules. Initiation begins when the beginning codon of the mRNA, which is AUG, attaches to the large ribosomal unit. The next step happens when tRNA with an anticodon. RNA's anticodon matches with the triplet on the mRNA, and the tRNA, where the amino acids that the tRNA was attached to, is from the tRNA. All the while, a new tRNA codon of the mRNA and the new amino acid that is detached connects with the previous amino acid, aim for a polypeptide. The peptidyl is what connects the amino acids together to form a polypeptide. It joins together just like that. After the tRNA detaches from the amino acid, it translocates again to the E site where it is done. Then, termination happens when the ribosome encounters 1 out of 3 stop codons. The completed polypeptide, the last tRNA, and the ribosomal units are then released.